How did life originate on Earth? Most of us were taught in school that life on Earth originated from the so-called primordial soup. In this soup, there were chemical reactions that made amino acids, which form the proteins. And the result was the first single celled organisms appeared. Nobody has ever seen this happening, nor any experiment has ever demonstrated it. This idea goes directly against the law of biogenesis, which states that in nature life only comes from life and from its own kind. In the 1950s, Stanley Miller was able to synthesize amino acids by passing electric sparks through methane gas. Although amino acids are not alive, this has given the hope to the evolutionists that one day they will be able to create life themselves. Proteins are made out of amino acids, and since proteins are found in the living organisms, the evolutionists were hoping that they would be able to explain how the origin of life on Earth happened by chance. What are the odds for this happening? Egling Louis Hashbeck answered this question saying, to make a simple bacterium requires approximately 20,000 enzymes. Enzymes are simple collections of amino acids which have come together in a particular order and structure. The structure of an enzyme allows it to accomplish a particular task needed by the bacterium. Since the number of amino acids and the structure of enzymes are now known, we can calculate the odds of forming the enzyme needed to create a single bacterium. Now listen to this. The odds of obtaining a single enzyme with a proper function is 1 in 10 to the 20th power, which is 1 followed by 20 zeros. The odds for obtaining all 2000 enzymes needed to create life would therefore be 1 in 10 to the 40,000th power. So this is 1 followed by 40,000 zeros. This number is so large that is beyond our comprehension. And that is only the beginning of the problem. Even if we did get all of the enzymes, most of enzymes are proteins, we would still need to get the DNA, which contains genetic instructions for the development, functioning, and reproduction of all cells and organisms. The following is the summary of the presentation from geneticist Professor Dr. Tomislav Terzin. The title of YouTube video is Genetički kod. We have between 50 and 70 trillions of cells. In the cell there is a nucleus where we find the DNA. The DNA is a molecule that has genetic code program in it and tells the cell how to make a human being. We would expect that if we came through the evolution, that the technology that we see in bacteria is less developed technology than in a butterfly, which is less developed technology than in a bird, which is less developed technology than in a human being. But that's not the case. From bacteria to human, there is the same genetic code, the same 3D printers, the same organelles within the cells. Every living thing has a different complexity, but at cell level we are practically dealing with identical technology. Second, if the genetic code was created by evolution, why we cannot see progress? In human technology, we see progress when we compare today with, let's say, 30 years ago. However, there is absolutely no progress at the molecular level. It doesn't correspond to the evolutionary predictions. If it doesn't correspond to the evolution, then where does it fit? For the technology that we have in our cells, aliens are not enough. We need nothing less than God. How is the DNA molecule copied? DNA cannot copy itself. You need to have molecular machines, proteins, that will make two copies of DNA from one copy. Do you know where the formula for making these molecular machines is written? Inside the DNA. You cannot make a DNA without a protein, and without DNA you cannot make the protein because you don't have the necessary information. So what is older, the chicken or the egg? You have to have both of them from the very beginning. You need to have 5 or 6 different molecular machines to be able to copy the DNA molecule. If you miss one of these machines, you cannot copy the DNA. If you don't have that first machine to open the DNA, you cannot copy the DNA. If there is no machine that will keep separate strings, you cannot copy the DNA. If you don't place the primer that later you will have to remove, which does another machine, you cannot copy it. If there is no machine that will glue those parts together, you cannot copy the DNA. If you don't make the necessary proteins, you cannot copy the DNA. When you make a copy of the DNA in one direction, you can copy it without problems. But since the other side is in the opposite direction, the machine copies it in reverse. So two DNA molecules are copied in different ways. And you must have both ways from the very beginning to make two cells from one cell. There is no evolution of this technology. No simple copying in bacteria and more complex copying in humans. The same technology. When the machine finishes copying the DNA and comes to the end, it cannot copy the last few letters. However, we have a special machines that at the end of the process extend the DNA molecule in order not to lose information. And at the end, special proteins machines keep the DNA from unzipping. You see how complex it is? Something that the theory of evolution says is the result of accidents. And as if this is not enough, those genes that are written in our DNA are interrupted. You don't have one continuous DNA information on how to make a protein, but you have several letters that make sense, then you have one part that makes no sense, then you have the next part that makes sense, and on and on and on. You need to put together pieces that make sense to be able to make the protein, and you need to throw out all the parts that don't make sense. And all of this should be done without the small dwarfs in the cell. All of this must be done by the machines, by proteins. So we have finally made the RNA that now can travel to 3D printers and make one of 100,000 proteins that exist in our cells, and each of them has its own different job and task. But the problem is that one machine cannot do it all. We need to have 20 different machines to connect 20 different amino acids so that the 3D printer can work. The 3D printer will make a protein like a tape, but now you have to assemble it to be a three-dimensional functional protein. This takes place in another machine. Then comes the transport of the proteins. Some proteins will be needed in nucleus, and they have to go back through the pores to the nucleus. Some proteins will go to the mitochondria to make energy for the cell. Some will go into the cytoplasm, and some should be delivered outside the cells. All proteins have an address at the beginning, and there is a system that transports them to their proper place. In order for this to happen, you must have the roads, and you have to have couriers who will carry the proteins. They are special molecular machines that literally walk on two legs. But that's not the end. We have mechanisms for the DNA repair. The theory of evolution says that the mutations in the rewriting of the DNA are motors of evolution. Because if there are no mutations, there cannot be evolution. However, all living beings, from bacteria to human, have extremely complicated mechanisms to fight against errors, against mutations. TVs, washing machines, computers are all made by someone. The human body is incomparably more complex, so it's impossible that it was created by itself through evolution. God is the only one who could create the cells. We are going to continue this topic in part 2.